Vectors which accept larger fragments of DNA than phage A or cosmids have the distinctive advantage that fewer clones need to be screened when searching for the foreign DNA of interest. In addition, they have had an enormous impact in the mapping of the genomes of organisms such as the mouse and are used extensively in the human genome mapping project. Further developments have given rise to the production of large insert capacity vectors based on the bacterial and mammalian artificial chromosomes, which are called BACs and MACs, and on the virus P1 artificial chromosomes, which are called PACs. However, perhaps most significant is the development of vectors based on yeast artificial chromosomes or YACs, which could accommodate hundreds of thousands of kilobases each. YACs containing a megabase or more are known as mega YACs. YACs are linear molecules composed of a centromere, telomere and replication origin termed as ARS element. A YAC contains a left and a right yeast chromosomal telomere, which are both necessary to protect the chromosome's ends, and a yeast centromere, which is necessary for segregation of sister chromatids to opposite poles of the dividing yeast cell. The centromere is placed adjacent to the left telomere, and a huge piece of human or any other DNA can be placed in between the centromere and the right telomere. So here you can see this is YAC plasmid, and it contains, this is a centromere, this is origin of replication, and these are two telomeres. So this plasmid is cut by BAM H1, so it acquires this form, and here in a fragment of DNA can be added. So after addition of a foreign DNA, it will become like this. The large DNA inserts are prepared by slightly digesting long pieces of human DNA with a restriction enzyme. The YACs with their huge DNA inserts can then be introduced into yeast cells, where they will replicate just as if they were normal yeast chromosomes. Using YACs, geneticists made great strides in the mapping phase of the human genome project. They produced a genetic map of the whole genome that provided an average resolution of 0.7 centimorgan. A centimorgan is the distance that yields a 1% recombination frequency between two markers and corresponds to an average of about 1 MB in humans. These researchers also produced relatively high resolution physical maps of two of the smallest chromosomes, that is chromosome number 21 and Y chromosome. These maps were especially useful in that they represented long stretches of overlapping DNA segments cloned in YACs. Thus, in the days before the human genome was sequenced, if you were interested in a disease gene that mapped to one of these chromosomes, you had a much simplified task. You needed only to discover two markers flanking the gene of interest. Look on the map to find which yak or yaks contained these markers, obtain the yaks, and begin your final search for the gene.